face when we think about regenerative medicine is that these are just raw numbers of deaf people around the world. And if you see the main cause of death globally, but it's even more evident in Western industrialized countries, are non-communicable diseases. And when we think of the diseases that are causing death in the world right now, are things like diabetes, diseases of the cardiovascular system, respiratory diseases, diseases of the digestive system, etc. These are diseases where there is one organ or tissue that fails, but the body, the rest of the body, is not failing, and this is what causes the disease, and in the end the patient dies and ends up in this table, but this is preceded by years or even decades of a very, very low quality of life, and this is what we want to solve. So if an organ is failing and the rest of the world, or the rest of the body and the rest of the functions of the body are working, the ideal solution will come from this type of process. This is regeneration. This is what happens in certain animals when you amputate some parts of their body, in this case a limb, and they regrow the limb. They have the ability to regrow other structures, to regenerate the retinas, even the spinal cord, pieces of the eye, the heart, etc. We don't have this ability. So what we have been trying for the last years to try and come up with something that will do or will try to implement this job. We have been very successful in the past years at using something similar. We are replacing organs. This is in Spanish, because these data are taken from the Spanish Society, the Spanish Agency for Organ Transplantation, and these are, which is one of the most successful in the world. We can see that over, these are data from 2013, and we see that this is the number of transplantations that have been done from kidney, liver, etc. We see that about 5,000 people in Spain have been transplanted with an organ, and that has solved their problem. Again, we have learned many things from organ transplantation over the years. One of them is that it works. So we can transplant a kidney, we can transplant a liver, we can transplant a heart, and the problem goes away. The other thing that we have learned is that this is not enough. This is the ratio of transplantation. I said Spain is an example in this particular area, because we have achieved the highest ratio of transplantations of donors per population, but those ratios have reached plateau several years ago. We don't get any more donors, irrespective of all the measures that we have tried to increase the number of donors, and this is happening worldwide. So we can cure about 5,000 people in a country of 40 million people, where most of the people are dying because of this problem. So this is the other lesson that I think we have learned from organ transplantation, that it works, but it's not enough, and it will never be enough. So we are coming up with this type of general strategy that we call regenerative medicine. But what we intend to do is combine, these are the famous 3 pillars of regenerative medicine, when we try to combine strategies using some type of cells, some type of biomaterials that will provide scaffold and instructions to the cells and signals so the cells differentiate. And in this scenario, I will talk mostly about the cells. And this is an image taken from internet. You can find billions of this type of images of all the promises and all the problems that will be solved using stem cells. Before getting into which ones of these may hold true or may be just a fraud, I want to introduce stem cells and what we consider stem cells, how they are defined. And for this, I would like to introduce a couple of concepts that we use that basically define stem cells. One of them is differentiation or specialization. The other one is potency. So Specialization or cell differentiation is very easy to understand. Of course, if we look at our body, and in our body there are many cells, and many, when I say many, I mean many. There are many cells in our body, and they come in many different flavors, many different shapes, sizes, functions, etc. So there are about as many cells in one body as vertebrates. All the living vertebrates on Earth put together, there's roughly the same number. But if you think of how you came to get all these animals on Earth, all these vertebrates, that takes hundreds of thousands of years of evolution. And our cells, they're different in two basic scales. One is time. We differentiate all our cells in our body, or the basic types of cells, in a matter of weeks instead of hundreds of thousands of years. And in evolution, we have a driving force, which is mutation. And this diversity is accomplished by mutating the DNA and selecting mutations. In here, our diversity of cells is accomplished using the same genome. There are no mutations in the genome during this process. So we have to play with epigenetic modulation of the expression of these genes and not the gene itself. So those differences are important to keep into account. And when you look at this process, this is of course accomplished during embryo development, and there are several things that appear or emerge during development. One is cell specialization. You also see patterning and the formation of different structures. But there is something that we normally don't pay attention to, and is what is lost. And what is lost is precisely potency. We define potency as the ability of a cell to, when it divides, give rise to cells different from themselves. And the potency is, of course, maximum at the zygote. But our cells, our somatic cells in the body, most of them have no potency at all. So this is a basic principle of embryo development. That is, a developmental potency decreases as development proceeds. Okay. So once we define these two concepts, the stem cell, or what we call a stem cell as biological unit, is a cell that is able to undergo unlimited self-renewal and keeps a degree of potency. So when it divides, it can give rise to cells that are different from itself. It can be a large degree of potency, such as the one that you find in embryonic stem cells or in pluripotent cells. And if you have a pluripotent stem cell, it will be a cell that is able to divide indefinitely and give rise to any cell type in the body. You can have a degree of potency that is less than pluripotent, could be restricted to a specific germ layer in the embryo. It can be just mesoderm, definitive ectoderm, definitive endoderm, and those we call multipotent. And depending on the source, we many times divide cells between embryonic and adult. I think that's easier understood if you think again on the way development proceeds, and if you take cells from this part of the development where cells are still pluripotent, they are not stem cells. Of course, we don't have embryonic stem cells, they don't exist in nature. But if we take pluripotent cells from an embryo where they still haven't to any of the germ layers, we can adapt them to grow in vitro. We can make them self-renewing entities, and those self-renewing entities will be pluripotent, and this is what we call pluripotent stem cells, and normally we obtain them from pre-implantation embryos. Or we can take cells from the adult, cells that are in many of our organs and are the ones that are responsible for the turnover of the cells in our organs. And some of them have stem cell properties in the sense that they can be maintained for the entire life of the animal dividing, and they typically give rise to the cells that comprise this organ. Okay. So go back to the organ transplantation. Another thing that we have learned is that when we transplant a heart, a kidney, a liver, or an intestine, the success of this is because we transplant the organ, but we also transplant the stem cells that maintain that organ throughout the patient's life. Okay? So this is where it came the notion of I may just transplant stem cells, and the stem cells will reform an organ and maintain the organ throughout the life, the patient's life. And this is where this entire notion comes from. And if you take stem cells from the brain, you could repopulate or regenerate a brain. If you take them from teeth, you can regenerate the teeth, etc. All of these are promises in practice. There is a basic problem using this type of technology, using adult stem cells, is that we don't know how to expand these cells. These cells exist, some of them exist. We know because we can test them in animals. We can test that biologically they exist, but we cannot grow them outside of the body. So we cannot expand them, and we cannot generate stem cells in numbers enough so they will be clinically relevant. We can only use this type of strategy in 3 areas at the moment. One of them is, of course, bone marrow transplantation. We cannot expand bone marrow stem cells, but we can take the entire bone marrow or a fraction of it that contains stem cells and transplant it at that, and this will regenerate the entire bone marrow of the transplanted patient. Okay. We can do the same for skin. We cannot expand skin stem cells so they will provide skin for the entire population or for several patients, but we can grow it enough so we can cover some parts of one's own body. Okay? And still, this is not perfect. We can grow some part of the skin. We cannot grow hairs, or we cannot grow other structures in the skin, but just the interfollicular epidermis we can grow. And the other area where we can apply this type of strategy is the cornea. So we can grow, similar to the skin, we can grow ex vivo stem cells from the sclerocorneal limbal cells, and we can grow some parts of the cornea. This we can implant in a patient containing also the stem cells, and that will regrow. This is done in people that have suffered corneal burns. They will grow the cornea over the life of the patient. So these are the 3 applications that are presently implemented in clinical application of adult stem cells. I wanted to raise this here because, of course, everybody knows that this is not the case, but at the dawn of this century, there were several papers in reputed journals showing that adult stem cells underwent this type of what's called transdifferentiation, metamorphosis. Many terms were used at the moment, showing that they were more potent or more plastic than it initially had been thought. And you could take adult stem cells from the bone marrow, from the adipose tissue, from the mesenchyme, from different tissues, and they will differentiate into cells belonging to different germ layers. They will be, in that sense, almost pluripotent. They could grow mesoderm-derived structures such as muscle cells or neurons from the ectoderm or intestine cells. This, we know that is not the case, but somehow this notion has permeated many discourses, and the rationale of using this kind of stem cells to regenerate structures such as brain or ovary, etc., comes from this notion that has been shown to be scientifically wrong. And there is no doubt at the moment that this is not the case. But I just wanted to raise this because in some in some environments this notion is still present. We know that there are pluripotent cells, and those were described first in the mouse, obtained from pre-implantation mouse embryos, and were called embryonic stem cells, mouse embryonic stem cells. And those are the cells that we typically use in the lab to generate knockout mice. So you see, since they were described in early 80s, these are all the knockout papers, basically mouse knockout papers that were published. This is a long time number of publications, but you see that there is an exponential growth around, or starting in 1998, which is of course when the first human embryonic stem cells were derived by the Thomson lab. And this generated this entire promise that we could generate embryonic stem cells that will be growing indefinitely in the lab, hence providing an eternal source of cells that we can then differentiate into any cell type that we want. So this is the story that we've been selling for many, many years now, and as you probably know, this hasn't, except for a couple of clinical trials that are now ongoing, this hasn't crystallized in any treatment in clinical practice. Part of the reason for this failure is that not all the research that could have been put into this line of research was effectively put because of ethical problems in many countries. What we have learned finding alternative to this type of cell is that this very notion that I described at the beginning of the talk, that potency decreases irreversibly with development, this is not true, and we can now reverse this loss of potency, and we can induce somatic cells to go back into their developmental history and produce in the lab cells that are pluripotent, cells that are stem cells, and that they are basically indistinguishable from embryonic stem cells. And this is what was discovered by Shinya Yamanaka in 2006 in the mouse, and what we now call induced pluripotent stem cells. So these cells grow and look like mouse embryonic stem cells. They have the ability to differentiate into all the cell types from the three main embryo germ layers, ectoderm, mesoderm, and endoderm. And in the case of the mouse, we can prove that from a single cell of this type, you can generate an entire mouse, which is otherwise normal, fertile. And that was 2006. The end of 2007, beginning of 2008, this technique was shown to be also valid for human. We can now take somatic cells, human somatic cells, reprogram them to something that is very much like a human embryonic stem cell. The way this is done is very easy. This is a very robust technique. It has been used in many species, of course including human, in many somatic cell types, and it works very reproducibly. It's just overexpressing a series of factors that we know which they are, because Yamanaka discovered that. And if you overexpress those factors over a very short window of time, the cell that you are trying to reprogram, in this case fibroblasts, undergoes a phenomenon called reprogramming. And in 4-6 weeks, in the case of humans, you get these cells that look like and behave like embryonic stem cells. So the use that we are doing, or the potential applications to this type of cells, are basically the ones shown here. Many labs are using these cells to understand how reprogramming takes place from the fundamental point of view. I will not go into details on this, just let you know that reprogramming, we have learned that is not a single-step process. It goes through different stages, and it involves a loss of the identity of the cell that you are reprogramming. Say you're reprogramming a fibroblast. The first thing the fibroblast does is forget it was a fibroblast, and it does so in terms of all the genes that are expressed. I mean, the transcriptome of that cell changes, but it's not until very late in the reprogramming process that the cell acquires an identity of induced pluripotent stem cell, In the middle, there is a long window of time where the cell has no identity, or at least no transcriptional identity. And this can be used in the lab to impose additional identities. The other thing that we have learned is that reprogramming itself is a very aggressive process. It's very aggressive to the genome, and the cell that is reprogrammed, if you look at it in the middle of reprogramming, it contains many de novo mutations, many de novo copy number variations. Most of them are solved by the end of reprogramming, either by selection or correction, but some of them still persist. What we are using these cells for, want to use these cells for, is to generate cells that will be transplanted to patients having a problem. If you have a heart problem, then you can generate iPS from that patient, iPS cells, differentiate those into heart muscle cells and transplant them to the patient. Okay, the story that we were selling with embryonic stem cells, because these cells have not the ethical limitations of ESLs, human ESLs, we can generate them from any patient. We can generate cells for autologous cell transplantation. And we provided proof of concept that this can be done. We did this in a disease called Fanconi anemia, where these kids, the problem they have is that they don't have the ability to form new blood. They don't have blood stem cells. So our rationale for this was, if we can take these patients, and we know the mutation that causes this disease, so we can take a biopsy from these patients, generate iPS cells, somewhere in this process correct the mutation that these patients have, so the iPS cells will not have the mutation. If we differentiate the blood progenitor cells, they will not have the mutation and they will be disease-free. So we can think of putting this back into the patient and recover the hematopoiesis in these patients. So we show that all of these steps can be done at efficiencies that are relevant for clinical application, but we also identify a number of shortcomings that are preventing the clinical application of these strategies. Some of these shortcomings have been solved over the last 4 or 5 years, mostly these that were technical in nature, such as the way we made the iPS cells. Now we have ways to make iPS cells that are clinically compatible. The way we correct the disease or correct the mutation, now we have ways to do this in clinically compliant manners. Will not go into details. I'll be happy to elaborate on this if you have any questions. But believe me, this is the case. But we can do that in a way that is clinically compliant, but is very, very expensive. So generate a line of clinical grade iPS cells is about €2 million. So you cannot think of producing a line for each patient or using this type of strategies in an autologous setting. Nevertheless, there are concerted efforts around the world to generate a bank of clinical grade iPS cells that will be used for, or will provide coverage to most of the population used in an allogeneic setting. And probably you know that Yamanaka announced a couple of years ago that a Japanese bank was being created for this purpose. And last year it was announced that the bank will not only be Japanese, but other countries are joining, will be a multinational bank that will provide coverage to about 85% of the world population. Okay. Other problems that we have are, of course, as with any treatment that we want to test, is our safety. These cells, I said that they are extensively manipulated in the lab. They are created in the lab, in fact, and there is doubt that all these processes will introduce mutations that will be deleterious and mostly causing predisposition to cancer. So there has been extensive preclinical data, safety data on these cells, showing that there are ways to produce these cells and there are ways to differentiate these cells into cells that are relevant for a disease and then implant into an animal model that are safe and they are no more prone to cancer than embryonic stem cells. Okay? But now we have to test if this is also the case in humans. So as you probably know, there is already a clinical trial that began last year in Japan using cells differentiated from iPS cells for treating a problem in a type of blindness. And we are waiting for the results of this trial. If this trial shows that there are no problems or no safety concerns using these cells, there are many, many other clinical trials in the pipeline to use iPS-derived cells in many different clinical settings. The problem that we find finally with these cells is a biological problem, is these cells, iPS cells or embryonic stem cells, have the ability to differentiate into any cell type in the body, but we just don't know how to do it in the lab. So we have protocols that produce functional cells for four conditions. For the rest, we don't know. We can produce cells that look like the cells you want. In my lab, for instance, we work mostly in heart muscle cells. So we can produce cells that look like cardiomyocytes. If you go to any of our talks, you always see beating things. Yes, they beat, but we also know that these look like very immature cardiomyocytes that if you implant them into an animal, let alone a patient, they will be a source of problems. And that has been shown. This is recent results from the lab of Chuck Murray and Michael Aflam in Washington University. They are able to produce clinical-grade cardiomyocytes to treat this is already in monkeys, and cardiomyocytes persist up to 6 months. They solve the problem these are infarcted monkeys. They solve the functional problem, but they are a source of arrhythmias as well. So we know that the maturation status of the cells that we implant is not good enough. But many, many labs, as you can imagine, are working on solving this, and as soon as we have protocols that produce functional cells that will integrate into the patient's organ, this will be solved. And very briefly, I wanted to mention something that we have learned with these cells that was probably not intended at the beginning, but since we can make iPS cells from any person, including patients, we can also look at how the disease appears, and we can model the disease in the lab. And there have been hundreds of diseases that have been modeled so far in the last 4 or 5 years using iPS cells. So if you take a disease that is monogenic or with a very, very important genetic component and that is early onset or congenital, most chances are that it will be modeled very easily using this technology. Other types of diseases can also be modeled with more effort. And I'll give you just an example of Parkinson's disease, which is the opposite of a monogenic early onset disease. Parkinson's disease is one of the most paradigmatic examples of highly prevalent diseases. It's a disease that takes decades to evolve in the patient. It's an aging disease, and it's a disease that most of the cases are sporadic, and that happens for any prevalent disease that you can think of. Most of the cases are sporadic. There's no family history, but there are some patients that the disease do run in families, and when you look at the families, you can identify genes. In the case of Parkinson's disease, these are the genes that have been identified. So what we did was take patients of this disease, both sporadic with no family history and patients with Parkinson's disease associated to a specific mutation, several of each case, and generate dopaminergic neurons, which is the type of neuron that degenerates in these patients, and see if we could find any problem in those. And we see that those dopaminergic neurons obtained from iPS cells from these patients degenerate in vitro. And that gives you a model to study what's happening. And in the patients that had mutations in a gene, in this case it was a gene called LAR2 that is not very well known what it does, we could study what the mutated gene was doing in the neurons from the patients. And we could identify this in collaboration with Anna Corbett, Albert Einstein, that this LAR2 is a substrate of a specific degradation pathway called chaperone-mediated autophagy, and this is shared with alpha-synuclein, and when LAR2 is mutated, then alpha-synuclein gets clogged in the degradation pathway, and this is why it accumulates. So you can study this type of phenomenon because you have the disease-relevant cells, in this case dopaminergic neurons. This doesn't happen in other type of cells from the patients with the other genetic information from the patient. And we can, when we have this type of phenotypes, we can think on screening these cells for drugs or interventions that will rescue these phenotypes. But we have also learned something very important, is in addition to finding phenotypes that we see in the patients, to recapitulating what we see in the patients, what we are seeing is that what we generate using this technology are, in this case, dopaminergic neurons that recapitulate the dopaminergic neurons that the patient had when he was or she was born. So before any disease was present. So we can see actually how the disease appears. And we can identify things that have never been seen in patients, because when the patient presents in the clinic, he already has some signs and there's some degeneration. So we can see what precedes degeneration. And we can do, for instance, these are neural networks made in vitro from neurons differentiated from iPS cells. We can study these mini brains from the patients, and we can analyze very complex behaviors. So you see, we're just looking at how these cells interact with each other, and when one neuron fires up, how many respond, and we can really analyze complex behaviors. And this is one of them. This is called effective connectivity. So we can see this is neurons, normal neurons, how this network operates in this parameter. This is the neural network equivalent from the patient, and this is the same from the patient when we correct the disease. So this type of alterations, functional alterations, have never seen in patients, and we are hoping that we can predict the appearance of the disease using this type of information. So these are the three main uses. As I was saying, I see that I'm running short on time, so I would like just to finish, acknowledge the people that participated in the studies that I mentioned from the lab and the collaborations, the funding, which anyway doesn't compromise I don't have any commercial interest on these agencies or companies, and thank you for your attention.
Thank you, Angel. We are really short of time, but I would like to open the floor for questions for a few minutes, and I would like to actually start with one. Okay. Because, well, this was a very interesting overview of pluripotency and potential application. But in this context, most of the people in this lab, if not in this room, sorry, if not all, are involved in reproduction, in some aspect of reproduction. So although you hinted to it, I would like to ask you directly if in your experience, in your knowledge, are there any stem cell-based clinically proven therapies in the field of reproduction or reproductive diseases?
In my knowledge, I know there are some clinical trials using bone marrow or mesenchymal stem cell derived treatments for it's called ovary failure.
Okay. Ovarian failure, yes.
Ovarian failure. And I know there's some preliminary data out there showing I don't think anybody's claim at this point that the stem cells or the cells that you transplant will form the egg, but that they do something and prompt the ovary to mature oocytes. That I think is what the preliminary results from these trials are showing. There are more basic efforts into, for instance, using pluripotent cells, pluripotent stem cells, to produce oocytes or to produce well, germ cells in general. But I don't think there's any clinical, foreseeable clinical application for this at the moment. Thanks.
Professor Raya, I am connected with I'm a clinician, but in my university there is an active stem cell laboratory working in, I think in Parkinson's or Alzheimer's, looking at other areas. But I'm interested in the testis and to generate sperm. And I'm in discussion with my the stem cell laboratory. Now, should I be looking at giving them some cells from the testis when we do the testicular biopsy? Or should we be using cells from some other part of the body for autologous sperm production? Do you have any ideas in sperm production?
Well, definitely you should.
Should from where?
You should give them the biopsies.
From the testis?
From the testis and see what they...
And that's more likely to produce sperm cell rather than, say, skin cells? Fibroblasts.
I don't think they're incompatible. One other application that I forgot, of course, and that has been used is to try and expand spermatogonial stem cells, or to use these spermatogonial stem cells to further form pluripotent stem cells. So as far as I know, spermatogonial stem cells can be used.
Maybe I'm mistaken, but I think the question relates to memory. You know, like epigenetic memory. If it's better to use cells from the same tissue that you want to regenerate when you reprogram them.
So they are asking you for testis biopsies to reprogram?
They're asking me to advise them which is the best. Should we be using, finding some stem cells within the testis, or should we use iPS cells from some other tissue? Well, what is your recommendation?
It's to produce sperm. To produce autologous sperms, if you can produce them from spermatogonial stem cells that you can somehow isolate and expand from testis biopsy, that will be, of course, the best. There is a problem associated to that, as in the case of any adult stem cell, that there is not reproducible working techniques at the moment to do that. But if they can investigate and they can come up with a way to expand the spermatogonial stem cells ex vivo, that be, of course, the treatment of option. In the meantime, as an alternative, you can give them skin from those patients. They will produce iPS cells that then can differentiate into something similar to spermatogonial stem cells or to spermatozoids. But as I said, not only for this, but common to any other organ that you may think of, the ways we have to produce now functional spermatozoa from iPS cells are very rudimentary. So both problems need to be addressed, need to be solved, and there's research space to do in both.
Okay, so if there's no other question, I would like to thank you again, Angel.
Thank you.
And